Hi, thank you for joining us for this ECIS webinar dealing with cell attachment and spreading. My name is Charlie Keese, and I'll be your host for this approximately 30-minute long webinar. Sometimes the slides may update slightly behind the audio. Please just be patient if that happens. Let me begin by briefly introducing the idea of ECIS, electric cell substrate impedance sensing. The ability to monitor cells using weak AC currents was first reported by Ivor Gaver and me when we were at the General Electric Research and Development Center. There we realized it was possible to monitor cells electrically using very weak AC signals at different frequencies. The heart of the system, of course, are the special wells that contain the electrodes. In this next slide, you're looking at an 8-well, an array. You can see the 8 wells in the upper left-hand panel. And at the bottom of each well, there is a large counter electrode, which is simply used to complete the circuit. The active electrode is a small 250 micrometer diameter circular electrode. And so on that electrode, you can monitor from a single cell up to, in a confluent layer, maybe 50 or 100 cells. Of course, monitoring more cells is often desirable, and it's particularly true when we look at cell attachment and spreading, and we make a variety of different 8-well and 96-well arrays with different areas and different configurations of electrodes. Regardless, though, of the electrode system you're using, the measurement is much the same. We use tissue culture medium with serum, complete medium, as our electrolyte. A very weak AC signal is applied between the electrodes at the bottom of the well. Being an AC signal, we have a choice of a variety of frequencies from as low as 10 hertz up to 100,000 hertz. What we do if we monitor the voltage, we simply take the current that we're applying and we divide that into the voltage, and using Ohm's law, we come up with the impedance measured in ohms. But being an AC measurement, it's quite a bit richer. And that's shown in this little inset. We're applying a current at a given frequency, indicated by the red dotted line. And we're monitoring the voltage, the solid black line. Of course, these will have the same frequency, whatever we apply. But the important point here is notice that the two curves do not necessarily peak at the same time. There's a slight difference in their synchrony. We indicate this with a phase angle. And without worrying too much about how this is all accomplished, it allows us to break the impedance down into its components, which is, in this case, a pure resistance and a capacitance, which comes from the gold metal electrodes in solution, and it's affected by the capacitance of the cell membranes as well. So the output of the ECIS system is either the impedance or resistance measured in ohms, or the capacitance, which is measured in nanofarads. Let me show you just some typical data as we inoculate one of these small 250 micron diameter electrodes. Here we've chosen 2 different epithelial cells, BSC1 and NRK cells, and these are introduced shortly into the beginning of this run. We're monitoring the resistive portion of the impedance here using a 4,000 hertz signal. And what you see is as the cells attach and spread, there's a dramatic increase in the resistive portion of the impedance, going up almost 7-fold from that of an open electrode. The BSC cells attach much more readily than the NRK cells, but they both happen to come to approximately the same final resistance in a confluent layer. Very often, though, we'll see very tight layers of cells that would be off the top of your monitor, or weak cells that affect the impedance very weakly, such as fibroblasts, which may only double the impedance from that of an open electrode. Once the cells have attached and spread, you notice there are fluctuations in the curve. These are biological in nature. Notice the lower trace without cells is completely flat. So we're seeing movement of the cells even in the confluent layer electrically. The reason we see these signals is shown in this next slide. Again, the electrical current is being carried by ions that move up to the electrode and accept or donate electrons. We have a gold electrode defined by insulating films, and the cells come down and attach and spread over the entire bottom of the well. But the current is maintained constant. And now the current must find its way through the cell layer. And there are two different paths it can take. One indicated by the solid red lines, which is movement of the current through the solution passages under the cell and through the paracellular space between adjacent cells. But notice there's also a dotted line in this figure, and that's to indicate that current, being an AC current, can couple capacitively through the insulating cell membranes. We're not talking about current moving through ion channels, rather we're charging and discharging the sides of the membrane, allowing AC current to flow. So with that very brief introduction, let's consider now monitoring the dynamics of cell attachment and spreading, looking at different extracellular matrix proteins. First of all, when we work with these gold electrodes, we're going to coat them with proteins by simple adsorption. Usually we use approximately 100 micrograms per mil or higher of the protein of interest, and we adsorb this out of a normal saline solution for a few minutes. The gold is very surface active and will pick up a monomolecular layer of the protein. If you choose not to define the electrode with a known protein, of course, it will still pick up proteins from the serum or other proteins that might be in a defined medium. So the cells are always growing upon a protein layer, not directly upon the gold. In this case, we've inoculated cells, adding enough cells to go to confluence upon attachment and spreading, and the electrodes have been coated with different proteins. And it's very clear looking at these WI38VA13 cells, a cancerous type of cell, fibroblast, that fibronectin allows much more rapid attachment and spreading than laminin, and elastin is somewhat intermediate. You can also see a dotted trace here where we have not added any specific protein and are growing it on whatever proteins from the serum have adsorbed on the electrode. But again, you're getting back data on how rapidly the cells attach and spread without opening the door of the incubator or doing any sort of manipulation, just reading the output of the instrument. Because in this case, we're looking at one of these very small 250 micron diameter electrodes, if using 100 micrograms per mil of a very valuable protein seems a little excessive, it is only necessary to coat the small electrode in this case, because only those cells are being seen. The large counter electrode that completes the circuit has a very low impedance compared with this tight bottleneck in the current flow. Let's think a little bit about how frequency can affect things in this measurement, because as I pointed out, the ECIS instrumentation can monitor anywhere from 10 to 100,000 hertz. We see in these figures there are two different current paths that we mentioned before. The green solid lines are indicating current that couples capacitively through the cell membrane, whereas the red solid lines are indicating current that is flowing in the solution under and between the cells. Current will always choose the path of least resistance. The passage of current through the solution is not basically dependent upon the frequency, but the ability to couple capacitively through the cell membrane is much more effective at high frequency. So in the top panel here, at, for example, 40,000 hertz, most of the current flows capacitively through the cell membranes. If we turn the frequency down for example, to 2,000 hertz in the lower panel, that route now becomes more difficult, the capacitive coupling, and the current instead will flow under and between the cells. We can show this effect quite dramatically in the next slides that were done by Joachim Wegener at the University of Regensburg. Here he looked at the attachment and spreading of MDCK cells, and he monitored two wells at 40,000 and at 400 hertz. These are the same wells. The instrument is simply switching back and forth between these frequencies. What we see in the first panel is that about 3 hours into the run, the first blue dotted line, at 40,000 hertz, the curves plateau. But the same wells, notice, monitored at 400 hertz at 3 hours are barely off their starting value. And it's not until much later, 10 hours after inoculation, that we see these curves reaching their final values. So what is going on is easily shown by looking at some micrographs of these cells. Here, we've looked at these cells at these 2 points, 3 hours, the upper panels, and 10 hours after inoculation, the lower panels. And the cells have been stained with the tight junction protein Z01 or E-cadherin. We've also stained the microfilaments with F-actin. And what you see is that the cell layer is essentially in place and attached and spread at 3 hours after inoculation. But it's not until 10 hours after inoculation that we see the junctional proteins organized and the belted pattern of the F-actin. So what this means, going back to the ECIS data, is that by monitoring at 40,000 hertz at high frequency, we can monitor cell substrate coverage. At lower frequencies, for example 400 hertz, we can monitor the formation of the tight junctions. Again, these are the same wells, we're simply using different frequencies to probe different aspects of cell behavior. Our interest today, of course, is in cell substrate coverage. It turns out the better way to monitor cell substrate coverage is to not use resistance or impedance, but rather to look at the capacitance, which is always being measured by the instrumentation. The gold electrode, as you see on the left, is a large capacitance. It's about 10 times as large as the capacitance of the cell membrane for the same area. As the cells attach and spread, and we go to high frequencies, so the current is going to couple through the cells, now it is coming out of the gold electrode and coupling capacitively through the cell membranes, which have a much lower capacitance, about 10-fold lower than the capacitance of the gold. When capacitors are connected in series, as we see here, the capacitance can be no higher than the lowest capacitance in the group. And so as cells attach and spread, whereas they increase the resistance, they will decrease the measured capacitance. And this is particularly striking if we go to high AC frequencies. So let me show you an example of this. Now we're going to follow the capacitance measured in nanofarads as a function of time when we inoculate MDCK cells on adsorbed fibronectin. The other aspect of this experiment is that the green traces are with only calcium as a divalent metal ion. The black traces are only with magnesium as a divalent metal ion. And we see that in both cases, although there are subtle differences, the cells attach and spread regardless of the type of divalent metal ion present. On the other hand, if we were to look at the tight junctions, again, using a 1 kilohertz signal, we see that these same wells only show this increase in the impedance, now in this case, when we have calcium present, which is well known for tight junctions. The beauty of using capacitance is shown in this slide. Here we're seeing the capacitance decreasing upon MDCK cell inoculation. And again, we're adding enough cells to go to confluence upon attachment and spreading. At the 4 different time points, the cells have been looked at with interference reflection microscopy to monitor the degree of cell coverage. And what turned out from these experiments is that by using capacitance, it will tend to be linearly related to the degree of surface coverage. So we have a means using high-frequency capacitance measurements to effectively do image processing and talk about the degree of cell coverage of an electrode. Let me show you some data now with different proteins. Here again, we're using MDCK cells. We have adsorbed either fibronectin, laminin, vitronectin, or bovine serum albumin on the electrodes. At time 0, the cell suspension is added, and now we watch the capacitance drop much more rapidly for fibronectin than for the other proteins, bovine serum albumin, requiring the most time. We're seeing these cells, in other words, going from a cell-free electrode to a confluent electrode. And because of this relationship we just talked about, we could replace that axis with the percent of cell coverage. And as you can see, we go from 0 to 100% here. We could also talk about the time it takes for these to reach half coverage, or the slopes of these curves over a given time, which are shown in the small insets. So this is our preferred method to monitor the dynamics of cell attachment and spreading. Let me show you some other data using the same system here, but a different experiment. Here we're again looking at MDCK cells that are inoculated on fibronectin-coated electrodes. But in the medium, we have different concentrations, which you can see listed there, of the RGDS peptide, which is going to competitively compete for the recognition site that the integrins are seeing on the fibronectin that's adsorbed on the electrodes. And we can see we can delay the rate of cell attachment and spreading by adding increasingly high concentrations of this RGDS peptide. If we do this exact same experiment, but we change the coating on the electrode to laminin, this is what we see. Again, we're not competing in the same way with laminin as we did with fibronectin, and the cell attachment and spreading is effectively not affected by the presence of the tetrapeptide. Here's another experiment. Here we've adsorbed bovine serum albumin on the electrodes and inoculated them with MDCK cells. And you can see the response indicated by the solid lines as the cells more slowly go from an open to a confluent electrode. Next, we then take and trypsinize the cells with mild trypsinization after they've been in place for 24 hours, and then re-inoculate these same electrodes again with MDCK cells. And notice now the dynamics is much increased as the cells attach and spread on whatever the cells have modified the electrodes. And if you look at these actual values, it appears to be a laminin-like response, which is one of the major extracellular proteins that is put down by MDCK cells. One other topic, very briefly, we have a whole webinar on using ECIS to do cell migration experiments using wound healing. But let me just point out how we differ from a traditional scratch wound healing assay. For example, if you had coated your dishes with fibronectin and then remove a layer of cells to look for cell migration, for example, with a pipette tip, you will also damage the fibronectin that's present on the electrode. And in those open spaces, serum proteins from the medium will adsorb. And so as the cells now migrate in to heal that wound, and ultimately close it up, we're not looking at migration on fibronectin directly. With the ECIS wound healing assay, we electrically wound the cells sitting on that small 250 micrometer diameter electrode. And then we can monitor the dynamics of migration as the cells come back in to heal the wound. Again, we're using capacitance at high frequency, which is our choice to look at coating of the electrode. And we're working here with the human prostatic cell line DU145. Electrodes were pre-coated before this experiment with either BSA or gelatin. And at time 0, we have a confluent layer. We apply a high current pulse to kill most of the cells on the electrode. And then we watch as the capacitance returns back to low values as the cells move in. And what we see clearly is that on BSA, the cells are migrating more rapidly than on the gelatin-coated electrodes. Well, that's what I basically wanted to cover today. The ECIS V theta instrument was used for all of the measurements you saw today. This comes with either a device in the incubator that supports 2 8-well arrays or a 96-well. And this part of the group that's shown on the right-hand panel is actually in the tissue culture incubator. There's another instrument we're just introducing, which is called the Cell Population Monitor, the CP96. This is a less expensive instrument which monitors cell capacitance and reports that data as the percent of coverage of the electrode. A complete turnkey system here is about half the price of the ECIS Z theta and sells for less than $17,500 in the United States. Its data will look something like this. You notice on the vertical axis, we call it cell X. This is the percentage of cell coverage. And in this case, we're looking at attachment and spreading, I believe, yes, of BSC cells starting at time 0. They form a confluent layer. It looks like maybe 20 hours into the run. And then they're exposed to different concentrations of DMSO. And we're seeing the loss of cells and rounding of cells upon the electrode. Just something you might be interested in knowing more about. Our website is biophysics.com. If you go there and click on publications, it will take you to a list of publications arranged by topic. If you click on attachment and spreading, you'll get a long list of publications that deal with the use of the instrument to monitor the dynamics of cell attachment and spreading and the effect of different protein layers. If you'd like more information about these instruments, here are some key people to contact. We'd also be happy to provide you copies of this PowerPoint slide presentation for your use. You can contact anyone here or you can contact me. I'm the last email address there, Keese @biophysics.com. This will be the last webinar until the fall. We pick up again every other Tuesday, starting September 22nd, where we'll talk about the use of ECIS to do toxicity measurements. And we hope you'll consider joining us for future webinars. I'll again leave these names of the key contacts on the screen should you wish to contact anyone. And I want to thank you for joining us today and hope you'll contact us should you have any questions, want information on demoing the instruments. We always insist you demo before you buy to make sure it does fit your needs. And we look forward to hearing from you. So thanks for joining us, everybody. Stay safe. Bye-bye.